Hello guys, it's your Omi here. In my first ever video, I'll be talking about enzymes, their functions and classification, terms like coenzymes, cofactors, cosubstrate and so on, like what they are and all. You guys can also take notes for your exams. So, let's get started. You might get asked in your exams, what are enzymes? Enzymes are biological catalysts. Means they speed up a biochemical reaction by lowering their activation energy. So activation energy is the amount of energy which is required by a chemical reaction to start up. It's like the starting energy for every reaction, because every chemical reaction needs a starting energy to get going. So the x-axis shows us here the amount of energy, and the y-axis shows us the flow of an enzyme activity. The blue graph represents the required starting energy or activation energy with enzymes, and the white graph represents the acquired energy without enzymes. The enzymes after a reaction remain chemically unchanged, means that they react with the substrate and convert it into products and go out of the reaction unchanged. This means that after an enzyme has catalyzed a reaction, it can go and bind to a new substrate and catalyze a new reaction. This can be shown in the equation down below, which shows us that enzymes and substrate make an enzyme-substrate complex for a short period of time, which then breaks down into the original enzyme and the product. The activity of an enzyme depends on factors like temperature and pH, and also sometimes on the substrate concentration. Most of the enzymes have the optimum temperature at 37 degrees Celsius. pH, on the other hand, depends on which organ do they work in. For example, in the stomach, the pH is around 2 to 3, and in the intestine, the enzymes have the optimum pH around 8. So how do enzymes actually work? You might ask yourself this question. So enzymes work when a substrate binds to the active site of an enzyme. Active site is actually the place where a substrate can bind to the enzyme. So when a substrate binds to the active site of an enzyme, the enzyme then undergoes chemical changes, which actually makes the enzyme perfect, the active site of an enzyme perfect for a substrate to bind. And then the enzyme can catalyze that substrate into a product perfectly. In other words, substrate induces a change of shape in the enzyme. That's why this model is called induced fit model. Induced fit model is actually a modified version of a key and lock model, which stated that the enzymes are like locks in which a one specific and only one specific key can fit in, and key in this case being the substrate. But scientists actually consider the induced fit model to be the correct or the probable one. So how many types of enzymes are there? There are actually 3 types of enzymes. The first type is the enzymes which break down larger molecules into smaller ones. For example, the enzyme like beta-galactosidase, which actually catalyzes the breakdown of lactose into glucose and galactose. The second category of enzymes actually build up larger molecules from smaller ones. For example, DNA polymerase 1. We all actually know this enzyme. It actually builds up DNA strands from singular DNTPs. DNTPs are the nucleotide triphosphates, which actually are present as monomers during DNA polymerization. The third category of enzymes are the ones which actually change one molecule into another, and they do that by transferring the functional group of the one into another molecule. For example, the conversion of glucose into galactose, and these enzymes as a whole, as a category, are also called isomerase. For example, conversion of glucose into galactose. So before we classified the enzymes on the basis of their function, how do they work, and now we are going to classify them on the basis of what they're made of. So there are actually 2 types of enzymes: protein enzymes and holoenzymes. Protein enzymes are made up of only proteins, like only amino acids, and holoenzymes are made up of apoenzyme and cofactors. The apoenzyme is actually the protein part of a holoenzyme, and the cofactor is the non-protein part of that enzyme. which means that a holoenzyme is made up of an apoenzyme, a protein, and a non-protein cofactor part. And cofactors are divided further into two types. The first type are metal ions like iron or zinc ion, and the second part are the coenzymes, which are actually organic parts of that enzyme. It could be ATPs, NADH or NADPH, sometimes also a sugar molecule, but non-protein part. Organic but non-protein. Coenzyme is further divided into two parts, cosubstrate and a prosthetic group. So the coenzyme is called a cosubstrate when the organic part binds reversible to the active site of an enzyme, which means when the enzyme is needed, the cosubstrate binds to the active site of an enzyme and it detaches after the chemical reaction is done. And a coenzyme is called a prosthetic group when it is attached to the enzyme irreversible. It means it's bound, it actually binds covalently and it's not detached after the chemical reaction is done. Thank you guys for watching. If you want me to cover another topic, just let me know in the comment section down below. And I also want to let you guys know that I'm new at this, but I'll try my best to improve my video quality. Until then, keep supporting my channel, and I'll see you guys in the next video.